So I was going to show a quick demonstration of at least how I get some things to work with sedfit and sedfat. Mostly sedfit today, and I'll have to see if I can find my data for the other set of data. Well, anyway, I'll put that off for a second. Let me share my screen. If there's no questions, you can post questions in the comments in the chat box. You can otherwise see what's going on. I think when I share screen, I should share everything so you can see all of the windows that pop up. Right now, I have sedfit already launched, and I was going to avoid talking about how to get there and how to get sedfit to launch. Why will it not share me? There we go. So I'm running Ubuntu. So I'm running sedfit under Wine. sedfit is a PC-only piece of software. You can get it to work under Macintosh. My favorite way of doing that is with a piece of software that's free called Wine Bottler. It seems to work on my older Macintosh that I use, and it's a great way of getting Windows and PC-based software to run on your instrument, on your Mac. There is no Linux version of sedfit or sedfat. There is a Linux version of sedinterp, but we're not going to go over that. That's pretty easy to use, basically. You could figure it out on your own. Ultrascan, I still have to figure out. There is a Linux version of that. These are all free types of software. So hopefully what you can see now is this blank screen. So this is what happens when you launch sedfit. Sometimes there are a couple of windows, a couple of pop-ups that say things like, oh, I can't find this and I can't find that. I'm running, I believe, version, I don't know where it displays the version anymore. It's version 16, which should be one of the latest versions of sedfit. There's a couple of key things to running the software. You generally want to follow things across the first 4 or 5 different menu items across the top. So you load your data. You can usually skip copy right away. You can display the data in different ways, but then you want to pick a model. And most of the time we pick this model, the continuous CFS distribution. So let's load in some data. We'll pick this model. We'll set some parameters. We'll run the data and we'll fit the data. And I'll explain a little bit about what the difference is between the two. I mean, the short answer is that running the data helps fit elements of the data set to the model that are linear. And fitting the data fits the highly nonlinear parts of your model. And it's best to do that in 2 steps, partly because you can see how well the fit is representing your data when you just do the run, plus the run is much, much faster. And it gives you good starting values for things like the baseline and boring stuff like that you're not particularly worried about. The things you need to worry about in sedimentation velocity is the meniscus position and the frictional ratio. You will struggle with both of these if you're trying to fit your own data. So let's load some data. And I'll see, I have data that I have stored into, it's the same data set that I uploaded to the website. This is just BSA data that comes straight from an experiment done a long time ago. What you see when you load the data is that there's a bunch of these files, and maybe it's worthwhile for me to find a window. Oh my God. If we go to we can actually look at what this data looks like, I believe. It's not there. Bummer. If we were to go to load this data file, and now the window's escaping me. Cool. Usually with the data, you want to look at these particular data files. Oh my God. This is great. So you're getting the real full experience. We'll just try to load some data and see if we can get some fits and mess about with looking at the data. Those files that we were looking at, there are files that are labeled according to the time they were measured and what was done during the measurement. So if we look at this load new data files again, we're in the BSA directory by default. The very first scan for interference photograph in cell number 4. So the prefix of this old school MS-DOS type data file is what scan number, pretty obviously, 1, 2, 3, 4, 5, 6. If you go all the way to the end, you'll see that we have something like 120 scans. And each one is an interference scan. So the interference data and the radial absorbance data are done in 2 separate files. So you get 2 files for each quote unquote scan. In this case, we're only looking at the interference data. So you load up the interference data from 1 to, let's just load up everything and shoot for the moon. If you click the shift key, when you click, or if you hold the shift key down when you click, you can get all of the data. This pop-up comes up asking if you want to load all of the data files. My laptop is, it was probably worth $1,000 way back in 2010. So it's probably worth $100 now. I would not use too much more than 100 scans for most laptops, even today. You could probably eventually use 200 or 300 scans, but I would use that window that I just clicked on to divide up the number of scans you look at initially, because you will be fitting and refitting this data multiple times. I have a previous analysis in it, but I'm not going to load it this time, so you can see what it looks like if you just want to deal with things from the very beginning. This comes up as a time dilation factor. We're just going to leave it the way it is normally. It turns out Beckman, in their infinite wisdom, managed to screw up the timestamps on their data files in one version of the instrument. So the old instrument we have is an XLI, the timestamps are fine. The new version we have is an Optima, the timestamps are fine. But there was a time with the Proteome Lab instrument that they had a couple of years ago where they managed to screw up when they were timestamping their actual data. And it means that all your data looked like garbage. You know, you got the wrong S values, you got the wrong molecular weight, because things that were happening at, 1 hour and 10 minutes were really happening at 1 hour and 30 minutes. They were off by a lot. So this is how the normal scan looks. It's got a couple of different weird features. There's this top window, and in this top window, there's a couple of things to see. There's a red and green line here and a blue and green line there. The blue line, it looks like it's set pretty well to what I'm calling the bottom of the data. So if you remember what I was showing you pictures before, there's a picture of a meniscus where things look a little crazy. This is the meniscus here. We're going to take the red line and we're going to drag it over to the meniscus. And I usually put it somewhere in here. Now the meniscus is a problem because you can see that I could put it in a whole bunch of places and they all look more or less the same. And the meniscus position has a huge degree of effect on the molecular weight of your protein that you're predicting. So instead of guessing where the meniscus is, we end up actually fitting it. So we use the red line to set a meniscus. We use the blue line to set the bottom. In sedimentation velocity experiments, most of the time we're concerned with the position of the meniscus. In sedimentation equilibrium experiments, it's usually the opposite. We want to take care that we know exactly where the bottom of the sample cell is. These 2 green lines are the areas that we're going to be fitting data through. Now, you might think that you want to fit your data all the way as close to the bottom as possible, but there's a couple reasons why you want to avoid that. First of all, in this particular data set, I don't have a lot of information about what's going on down near the bottom. Second of all, there's a back diffusion component that's difficult to model. And so I usually have back diffusion turned off, and it's usually off by default in most versions of sedfit. It's just really computationally intensive. You can use it, but what you're doing is you're modeling diffusion of this large amount of protein down here as it diffuses back in this direction. And so that's probably all accumulating in this area where the concentration profile is reflecting back diffusion. At some point, what we're seeing now is just the regular sedimentation of the protein. None of the protein here seems to have felt this wall, is what I'm trying to say. And so it makes it easier to actually fit the data. So we're going to chop out this portion of the data. We're going to fit between these two green lines. The other reason is that up around here, the concentration of protein is so high that it's not behaving normally. And we're assuming that our protein is behaving like non-interacting particles. So we loaded our data. We're going to skip copy. We're going to skip display. We're going to pick a model. We go to continuous C of S distribution. That's our favorite thing. There's a lot of different models you can actually pick, and you're more than welcome to play with them. In the old days, a lot of times people would use this particular model, the LSG star of S, which is an old-fashioned way of analyzing data. It works really great if you have a computer older than mine, and I really sincerely hope you don't have a computer older than mine. Mine can actually handle the continuous C of S distribution, and it's a great way of starting to model your data. So just pick this for the start of your modeling sessions. When you pick, it'll give you some errors. Not an error, but it'll give you some comments. When you pick when you pick a model, the parameter window will change a little bit. And so now we have a bunch of constants that we need to add into here. This part in this box describes the S distribution that we're going to ask it to model over. Right now, I'm going to leave the frictional ratio at 1.2, which is a perfectly good starting model for a hydrated sphere. A sphere should have a frictional ratio of 1, but a hydrated sphere is more like 1.2, 1.15, depending on how you argue. I don't know the size distribution of my protein, so I'm going to just scan the S values. If your sedimentation profile, your C of S profile looks funky, you may need to expand this number. To make it easy on my computer, I'm only going to have it calculate 100 points between 0 and 15, if that makes sense. I usually leave baseline correction on, but it almost always results to 0. For interference data, you should have time-independent and refractive radial independent noise on, and I'll explain that in a little bit. We're going to leave fitting the meniscus position alone. And I'm going to change my F ratio to 1 sigma, which is 0.68, but you could try leaving it at 0.95. It allows a few more solutions through that are quote unquote acceptable when it's at 0.95. 0.68, I think works a little bit better. There are then a couple other parameters. I leave these two alone. I would not mess with them. These things you should know about your solution. So you need to know the viscosity of your buffer and the density of your buffer. For water, in this particular experiment, this was done versus water. The buffer density is like 1, and the buffer viscosity is like 1 centipoise. Now, tragically, it's listed in poise, so 1 centipoise is 0.01 poise. This parameter actually has a huge effect on how things sediment because of 2 reasons. One is that if you're doing things at 4 degrees, it's no longer 1 centipoise, it's about 50% higher. It's like 1.5 centipoise. And so everything sediments a lot slower because it's going through a more viscous field, if that makes sense. If your buffer's viscosity goes up, then your sedimentation needs to be accounted for. And so that can be tricky to take care of, but this actually does it for us. So these are buffer parameters. You can actually calculate them from sedinterp. I have a couple of pieces of equipment that let you measure them if you want to. Generally speaking, unless you're adding weird stuff to your buffer, these defaults are going to be really close to correct. This is the viscosity for room temperature solution, 20, 24 degrees, it'll be about the same. The buffer density, if you look up the numbers, you're just changing things in the third or fourth or the second or third significant figure. So it's not really changing much. You can actually play around with that, with setting different values and seeing if that changes your model. It won't. This number you should look up a little bit about. For those of you that have had 565, hopefully it's something that's covered. The partial specific volume is basically the inverse of the density of your protein. You could calculate it from sequence. Sedinterp will do that for you as well. If you know the sequence of your protein, you can calculate the partial specific volume. But at the end of the day, most proteins, their partial specific volume varies between 0.72 and 0.74. So it's another super boring parameter. We usually leave it at 0.73 and just don't worry about it. So now we've filled out the parameters here. Basically all I did was change the confidence value, which is just a personal preference, and I turned on fitting the frictional ratio. And that'll do a couple of things for us. Um, if you right click, you can draw a box around your data and you can actually zoom in on it. So here's our data, and it has a couple of interesting features. It looks like good data, but it's actually jumping up and down. And so there's a thing, if you look at the height of this black line versus this lower blue line, it's almost quantized on this y-axis in terms of fringes. So if you remember, I talked about constructive and deconstructive interference for the interferometer. If the algorithm that turns the fringe position into this y-axis value, which is in your data file, if it guesses that your fringe starts at 0 and then it comes along to a different set of data and it guesses that the fringe starts at a different value, it will have what's called jitter. And the jitter is usually quantized by values of individual fringes, 1 or 2 or 3. So you can see that the maximum of this data set seems to be about 1, and the maximum of these seems to be about 2. That will get corrected for in the fit. So what I did is I loaded the data, I ran through some parameters, I'm going to click run. It solves the Lamb equations. Remember that crazy differential equation I put up in the experiment or the presentation earlier? And then it's going to calculate a bunch of stuff for us. And it's going to complain. It complains a lot. Residuals close to the meniscus are significantly larger than elsewhere. That's true. I didn't fit the meniscus, and that's okay. The model problem frequently shows up in this region, includes the wrong position of the meniscus. That's probably what's going on. The initial concentration may be caused by misalignment of the cell. That's probably not what's happening. This, I think, is a good data set. These help messages become really annoying after a while, and it tells you how to turn them off. I usually turn them off, but after a while, you just kind of get used to them. The other help message is that it's reminding you that the distribution value at the max value is going up. This usually means that you didn't guess large enough. I decided to go out to 15S, and it's complaining that it's trying to fit something out here that looks like it should be another peak, but it's just starting at 15S, meaning I should set my maximum S value to something larger. I ain't gonna worry about it because it's little, because I know the answer for this one. If I fit the meniscus, sometimes this distribution problem will go away. If I want to see what predictions I have already, I can come down here under display and do show peak molecular weight in the C of S. So since I'm doing the C of S distribution, I have all of this interesting information. I have the data up here. There's fitted values going through this data. I have this particular funky grayscaled thing called the bitmap, and I'll explain that in a bit. And then I have the residuals for the fit. And down here is the model. And the model is telling me that if I put something in at about 60 kilodaltons, if I'm reading that right, and something about what looks like it might be a dimer, and maybe even a trimer, then I can see that I can create data that fits my data. I can create a model that fits my data best. Right now, the things you want to pay attention to are these values over here. We have not fit the frictional ratio yet, so it's still at the default value that I started. We're not even trying to fit the meniscus, so it's just sitting there. So data, model, parameters, hit run, it'll just go through the whole thing again and it'll pop up the data. It'll give me the C of S again to look at. Hopefully it won't take too long. And then when you want to start fitting your data, oh gosh, when you want to start fitting your data, you click fit. And this is going to actually take a little bit longer because now what it's going to do is it's going to start trying to change the frictional ratio, which is listed right here, in order to best fit the data. And the frictional ratio has a pretty big effect on the slope of, well, let's just say the frictional ratio is highly nonlinear. So as the algorithm for doing the data fitting is adjusting the frictional ratio, you'll have a very large effect on the quality of the fit. So it usually fits fairly quickly. I think this one fits to a frictional ratio that's like 1.3 or 1.27. We'll see where it ends up actually being at. It might take a little while to get there. Once we fit the frictional ratio, you'll see this allocating memory space. It'll do all this calculating normal equations, blah, blah, blah. It's fit a frictional ratio of 1.27. The RMSD is a little bit down. If I display the molecular weights, they've actually changed a little bit. Now it's telling me that My main peak of BSA is rolling in at about 66 kilodaltons. And since I'm running BSA, that's about what I expect. I think when I do my final fit, it ends up being a little bit smaller than that, and I'm not really sure why. So all I've done now is that in the parameters window, I've allowed the frictional ratio to fit, but I really want the meniscus to fit. And you'll notice I turned off the frictional ratio because my computer's slow. And like I said, the two parameters that are highly nonlinear are the frictional ratio and the meniscus. It's sometimes easier on the algorithm to fit one of them at a time until you've sort of hacked them into the correct place. They get fine tuned into the correct value, and then you can go back and refit the data with everything being allowed to be adjusted. That's usually the most rigorous way of finding the true minimum. Every now and then, when you do the meniscus position first, you will find that the meniscus gets wiggled around and you'll see different things happening. If you get tired of waiting for a fit, if it's just going on and it's taking half an hour and you've run out of whatever refreshing adult beverages you may be consuming, you can press the space bar and it will stop the fit at any time and show you as close as it got. So you can actually recover your data. Um, Again, it's just fine tuning the C of S data. The meniscus position has changed a little bit. If you right click and zoom in, we can right click and zoom in on this area, and I can see that the meniscus position has ran into the fit limit. Now this is something that happens often as well. I'm going to move these positions around a little bit. I'm going to say confirm. I'm going to move the fit limits too. And what that's going to do is it's going to allow me to reposition the red line relative to these dashed lines. These dashed lines here is how far I'm letting the algorithm wiggle the meniscus position. Out here is crazy land. The meniscus is not here. The meniscus is somewhere here where all of this crazy looking data is. So I want to force the algorithm to pick a meniscus position that best fits the data, but actually sits somewhere around here. I might actually scooch this a little bit to the right, but sometimes when you're doing the fit, the meniscus position will run into one of the limits and it will just stop. That's usually a bad sign, and you want to open up the limits on fitting your meniscus position and go back and refit it, which is what I'm doing now. Like I said again, if you get bored, you can just smash the spacebar and it will stop the fit. It will recalculate as much as it knows, and eventually it should give me a fit. So you'll notice we're fitting and refitting the data a bunch of ways. We've really only started exploring one particular model. And in this particular model, we've kind of already made some assumptions about things, partly because I know what's in this sample. It's just BSA, it's fairly purified. I bought it from the store. I ran it on the gel. It looks really nice. So my interpretation is that this main peak is probably mostly my protein is BSA. When I look at the molecular weight of the second peak, it turns out it's about twice the molecular weight of BSA as predicted. You'll notice something interesting too about that, and we'll get into that in a minute. The BSA dimer is rolling in at an S value that is about, that it's not twice as much as you would expect for the S value of BSA. So for this particular experiment, the S value for BSA looks like 4.6 or so. That means that you would expect the dimer to be about 9, but instead it's actually a good deal lower. And part of that has to do with the fact that the way things sediment isn't exactly the same based on how they dimerize. I'm not sure I could explain that better, but generally speaking, in terms of sed coefficient or C of S distributions, your dimer will be about 70% of what you expect it to be from the multiplicity of the monomer's sedimentation coefficient, if that makes sense. Does anyone have any questions on this so far? This is taking really long. It's annoying me. So I'm going to stop this and I'm going to just fix the meniscus. This is the sort of thing where you would want to provide yourself a refreshing beverage and do something else in a different piece of software if needed. If you double click on the upper window, it zooms out. If you want to see more about what's going on in the lower window, you can do the same thing. What's nice about zooming out in the upper window is that it tends to behave properly in the bitmap and the residuals. Residuals hopefully make sense. If you were to zoom in the upper window, you'll see that there is data from the interference data, and then there's a model that seems to be going through the data pretty well, all things considered. So we'll zoom out there. The bitmap is a color of or a grayscale that's denoting the difference between the fit and the model at every scan. So you see you've got scans from black to red. Those scans correspond to the first line of this bitmap and the very last row of pixels of this bitmap. And each row of pixels corresponds to one of these rainbow colored curves. The grayscale of the pixel corresponds to whether the data fit or whether the model is too high or too low. So when you see this streaky behavior here in your bitmap, it usually implies that it's having a hard time getting that slope right in the very middle of the data set. But we're just going to leave that for now. I'm going to turn the parameters on. I'll turn the meniscus position off. I will toggle back to the frictional ratio and we'll see how fast it takes to get a fit. But then we'll zoom in on the C of S data and we'll normalize the raw data. If you're trying to publish something, well, you've seen some of the figures of how this looks, and by all means look at papers to get a sense of what people do when they publish things. You don't always show every single scan here, although theoretically you should fit every scan that you have. This data set, though, because of the interference jitter, is ugly. So what we can do is we can go to display and we can subtract all systematic noise from the raw data. If you click on that, it gives you a little warning that it's about to subtract fitted noise from the raw data. But what it gives you is something that makes a lot more sense to humans. This is your raw data with the fit. You can even see how in this very first scan, it's having a hard time getting a perfectly good fit, although it's only starting to fit from the beginning of the screen line. If you want to zoom in on the lower part of this, you can zoom in on the dimer trimer area. And now here's our dimer at about 6.5 S values. I'm pretty sure it's dimer because if I display its molecular weight, it's about 2 times 66. And that's about 3 times 66. If you want to know about how much dimer there is, you can click this button that pops up and it gives you a plethora of information. The weight averaged S value is 6.65 Svedbergs, and the value corrected for 20 degrees in water is 6.681. My experiment is really close to water, so these two numbers are very similar. This SW value is what you would use when you're trying to do a fit yourself. It depends on what buffer you have. It depends on what temperature you're running things at. When people are reporting a Svedberg or an S value for the size of their protein, they usually standardize that S value so that as if you were doing an experiment in water. Not all proteins behave in water, but we can extrapolate to what the S value would be in water. And that means that if I do my experiment in 500 millimolar saline, and you do yours at 150 millimolar saline, the difference in density in our two experiments isn't going to matter. We should have the same S20W. So the 20 corresponds to 20 degrees Celsius. The W corresponds to aqueous or just water-only conditions for your proteins to solution. It's a standard, and this is the number you'd want to report, so this is a good thing to write down. You have an effective concentration of how much material you have, and it's actually shown graphically in red that the dimer is contributing to this portion of your curve. You have a bunch of other information based off of the expected molecular weight, the molecular weight if it was a compact sphere. We don't think it's a compact sphere because our frictional ratio is scooched up to about 1.3. That gives you a certain prolate oblate type radius, which you can sort of fit. If you're tired of that info, you can click OK. If you want to zoom out to see all of the data for this entire region, you can do that as well. We can turn on the molecular weight information and explore what's going on in this one. It seems a little light. It should be 66 kilodaltons, and instead I'm getting 64 kilodaltons. Maybe my protein's degraded. Maybe the fit's not perfect. Um, but either way, it's weighing in at about 4.5 Svedbergs, 4.5 S. Um, and so again, this is a number you would want to write down. The other number you'd want to keep track of is that while you're trying to finalize your fit, you would go to show fit information again, and you'd keep track of something like this RMSD, which gives you a value, a figure of merit for how well your fit is comparing to the actual data. Does that make sense to everyone? Are there any questions on any of that? So let's assume I have a decent fit. It turns out there's a bunch of different fitting options. You can pick different ways of running the adjustment of parameters so that you get a fit. I've been using the simplex algorithm. It tends to work by default faster. Marquardt-Levenberg, I believe, takes a little bit longer. What I should do is alternate between fitting both of those schemes. And eventually, once I've wrestled this into an appropriate final fit, I will have the parameters set so that I'm fitting both the frictional ratio and the meniscus at the same time. Once I've got that to work, I'll go to the fitting options under options, and I will alternate the algorithm of the fit back and forth until it feels like it's got itself a nice fit. So for whatever reason, that seems to have worked. I'll go back to simplex, save it as a default. We'll turn both of them on. We'll see if that actually gives me a fit that converges. It should be pretty close. Nothing much is changing, except the RMSD might be getting slightly smaller, and the frictional ratio in the meniscus position might be changing just ever so slightly. But this is the sort of stuff you want to do before you ever publish your model, uh, based off of your data. Once you've got all of this stuff fitted, you want to start thinking about publishing a picture. There's a couple things that I should mention. Are there any questions on that? So this is like what I would call fitted data at this point. If you want to get the results of this fit into like a text file that you could load into your favorite graphing software, that's always an important consideration, and I should probably show you how to do that with sedfit. I'll just stop the fit now since we seem to be kind of close. Although I should really, if I had time, I would let it run to convergence. If you go to copy, you can do things like copy the data table, the fitted data, the residuals, the noise values, or this one, which I use almost all the time. Copying the distribution gives you a copy of a text file which describes this lower plot. So you could actually recreate this lower plot, which has your C of S model that describes the fit to this data, and you can plot it up in something else that you use for making publication quality graphics. Because most people don't just take a screenshot of their sedfit and then dump that into a paper. It's kind of functional more than it is pretty. So you can copy any of these parts of your fit, make it into a file, put it in Excel, load it up into Origin, plot it in GraphPad, whatever you like. The other thing that a lot of times people will do is, and I'll see if this actually works, you can save your distribution plot for a bunch of different files, but if you also install a piece of software called Gussie, then Gussie is basically for gussieing up your C of S distribution. It's written by a different faculty member, Professor Brautigam, I think of, what is this, Texas Southwestern. So he wrote something that lets you make nice pictures out of your C of S distribution. And what's really cool about it is that you can actually save your figure or save the form of the Gussie state, and you can do different things to your C of S distribution. You can overlay 2 or 3 different C of S distributions. You can change the color. You can make the fonts look different. And then you can save the figure as something that's scalable vector graphics or just a PostScript or a PDF. And that way it makes it a lot easier to incorporate pretty graphics into your final paper. You will notice when I was doing my fits previously that I picked 100 scans and parameters. So you might not notice it, but if I want to use the same fit but with smoother data, I might pick 200 in the resolution. And then when I go back to Gussie, I can load in that data and it'll probably take care of this sort of like pixelated data point here, if that makes sense to people. So you've got your monomer, your dimer, your trimer for BSA. Um, but there's a kind of, it seems like the monomer is very narrow in however the algorithm fit. But if I put more data points into it, it might come up with a smoother fit in the Gussie state. The Gussie has a lot of information in it that you can figure out on your own. It's all pretty obvious. There's a lot of scaling offsets. You can play with the legend and reorder the the painting of the different C of S distributions. But I wanted to point it out because in order to go from data to publication, Gussie is super, super convenient. You don't have to worry about saving the C of S data file. Um, and what's really funny is that it complains when you close it. So now I have a higher resolution C of S file. These curves should now look a little bit smoother because I put 200 data points between 0 and 15S instead of 100. All it did is it makes this part smoother. Does that make sense to everyone? So I'm going to zoom this out. Um, I wanted to show you guys one more thing that we do all the time, and someone was asking about this. Where the heck is it? It's Control X. Oh yeah, size distribution. So I was talking about this a little bit. If you know that you have whatever species, well, this is a little crazy that it thinks it has this many species, but we think that there's a species at 64, 114, and whatever this one is. Um, 150, 160, it's hard to say. It gets really ugly down here, I guess. I have too many data points. Maybe I should smooth it out again. But anyway, if you zoom out all the way, if I wanted to assert that BSA is not glycosylated and it doesn't have any polydispersity and it should be, 66,034 Daltons exactly, with enough deviation except for maybe it doesn't matter. The easiest thing to do is to go to size distributions, options, and use prior knowledge of discrete species. And all this is saying is that I want to remodel the data using these starting values and saying that there should be something that's 60 something kilodaltons. It should be infinitely thin though because it's 66,043 daltons exactly. And there's no distribution to it. And if it works, what you'll see is that the model in the lower panel looks like it's got just a line plotting the fitting the data. So your C of S distribution doesn't have any width to the peaks. The peaks are just a line. Um, if it doesn't work, then the C of S distribution has to sort of backfill in these little bumps near the line. And we'll see how that looks because I suspect it won't work perfectly with this one. Although now we're refitting the data with all of the parameters checked. So it's going to take a little liberties in that it will start with this initial value. It will give you a little report that it found two peaks um, of basically infinite thinness in terms of the resolution that I gave it for the S value. One peak at about 4.5 and one peak at about 6.5. And the two peaks account for a total amount of signal pushing 70% or above 70%. So what this curve is telling me is that we're getting an RMSD that's still good enough to model all of our data. And I'm using a model where there's a thin single molecular weight for this species and a single thin molecular weight for that species and nothing else. There are a couple other little divots and bumps in here that are showing up, but they're not contributing much to the actual overall signal. If you look at the contribution here, it's 1% of the total signal is contributing to this tiny little blip at 196 Daltons or kilodaltons. So maybe there's trimer there. It's hard to say. I don't know why it split this into two different species, but it does make me feel good that it's actually fitting most of my stuff to a single discrete molecular weight, and it gets a perfectly good fit, which implies that I'm not seeing the effect of glycosylation on this monomer or polydispersity. It could be that your protein was not properly made, the bacteria only made part of it and it just bombed halfway through. It could be a protease has been chewing on your protein. It could be you've got like two different plasmids, one with a stop codon in the wrong place. There's all these things that would give you some polydispersity in a natural occurring protein. Most of the time, though, the width of this peak is not associated with any polydispersity in proteins. Does anyone have any questions on that? To get back to your original plot, a lot of times you just hit run and it'll refit the data without the prior knowledge of the fact that your protein is monodispersed. And of course, because I have 200, a resolution of 200, it gives you a set amount of data. Now, when you're doing your fit, you should make sure that you toggle between the different fitting options. You should toggle between different fitting ranges. So an interesting question that you should ask yourself when you're trying to model this data is, does it matter if I move this green line a little bit to the left or the right? And you should do that and refit the data and convince yourself that it doesn't change the molecular weights that you're predicting. Does anyone have any questions? I see nothing. So I'm going to end it a little bit early so I can get things set up for a quick demonstration on how to load things. I'm going to try to assemble a cell, and I brought some samples. Where did I leave my samples? I think I left them in the lab. But I'll bring my laptop into the lab in 348, and we'll look at the old XLI. I think it's summer we'll go over the new instrument. It'll give me a little bit more time to get used to it. Like I said, we just installed it. But if anyone's interested in using it, by all means, we can talk about that as well. If there's no questions, I'm going to stop the recording and get ready for